Okay, hi everybody. Today we're going to continue with metabolic alkalosis, which we've been doing for the last number of weeks. As I often say, it's the most complicated of all acid-base disorders because of the different phases that are involved in its pathogenesis, and also because of all the different factors that either have been hypothesized over the last number of years to be causally related to the disorder, and to dissect from those hypothesized factors which ones are actually true. And the problem is when you're seeing patients on the ward or you're reading textbooks, it's extremely confusing because there's just been so many factors over the years that have been invoked. So what I've been trying to do the last number of weeks is to look at the actual data so that when you graduate, you'll have a deeper understanding of the pathogenesis when you're looking at patients. So this slide just summarizes a number of the factors that have been hypothesized. Starting at the first row on the left, hypokalemia and potassium depletion, they really haven't been distinguished, but you should conceptually. So this refers to the concentration in blood, and this refers to whole body balance. Is there less potassium in total in the body or in a given compartment, blood, ECF volume, intravascular volume, etc. And then on the upper right, extracellular fluid volume. We've heard often of patients are volume depleted in metabolic alkalosis, and some people say that giving volume corrects the metabolic alkalosis. As we talked about, and we'll talk about again, that is not true. Mineralocorticoids have been invoked as a cause. CD refers to the collecting duct. We'll talk about the anatomy in more detail today, but this refers to the concentration of sodium in the urine side, in the lumen of the collecting duct, as opposed to sodium depletion. So unlike potassium, no one talks about the serum sodium concentration like we do with potassium. What's referred to here is the collecting duct, or the urinary sodium concentration. But again, there's also whole body balance and potential sodium depletion. A lot of people feel that if you give sodium, the metabolic alkalosis corrects, or if you remove sodium, it's created. And again, as we talked about, and we'll talk about in more detail today, that is not true. And then there's the collecting duct chloride concentration, which we'll talk about in some detail because this is now felt to be extremely important, versus chloride depletion, again, whole body input-output. And then the GFR, we talked about the GFR a few weeks ago, and I'll talk a little bit about that again today. So metabolic alkalosis, remember, before you know anything about the pathogenesis, you have to clinically divide it up into 3 phases, because without thinking along these lines, you're going to have a blurred understanding of what's going on. The first key question you have to ask in any patient, whether it's someone vomiting, someone with Gittleman's or Bartter syndrome, no matter what the cause, is you have to ask yourself, where's the bicarbonate coming from? And if it's endogenous, then you're asking what vein is pumping bicarbonate into the blood. And at least answering that organ-based question will allow you to think more along the lines of therapeutically where you have to attack the system later when you're at that phase of looking after the patient. But generation or an IV of bicarbonate in a normal human being, and this is a very important concept, does not raise the bicarbonate concentration if you have a normal GFR. The kidney and a normal proximal tubule, the kidney just dumps the bicarbonate. You have to go to extremely, extremely high inputs, which we just don't see endogenously. You could do that with an IV, I guess. But endogenous bicarbonate inputs just are able to be dumped very easily by the kidney. So when you have an endogenous bicarbonate source and the kidney isn't dumping it, you have to ask why it's not doing that, because normally it would. There's something in addition to the input, the IV, endogenous IV input, whether it's the gastric vein or the veins coming out of the colon or the renal vein, Gittleman's or Bartter syndrome, whatever the vein, that input should be able to be dumped. If you started an IV with that amount of bicarbonate that a vomiter generates in the gastric vein, in a normal human being, they would just dump the bicarbonate. So something else is happening in the vomiter. Well, it turns out the kidney is what is going on. There's impaired ability of the kidney to excrete that bicarbonate load. And that has a name for the phase. It's called the maintenance phase. And that has its own pathophysiology, which you have to understand, because you're going to be attacking that problem when you're trying to correct the metabolic alkalosis. And then there's the correction phase, which can involve either attacking the generation or attacking the maintenance, or both therapeutically. And then when the bicarbonate does drop in the correction phase, you have to understand that also. There's pathophysiologic mechanisms involved, which in response to your therapy, whatever it is, cause the bicarbonate to decrease. And usually it's bicarbonate being dumped by the kidney. So the urine bicarbonate excretion rate goes up significantly. Okay, so practice thinking of these three phases and understanding the implications for each of them when you're seeing patients on the ward and you're treating them. Now, we talked about potassium as the first factor. Again, we didn't distinguish hypokalemia from potassium depletion, whole body potassium balance problems, just because no one has in the literature. No one has ever kept one factor constant and changed the other. It's just, you can't do it in animal models or clinically. So I'm lumping these together. But probably it's the hypokalemia because of the following. This picture here is to tell you that the mechanism for the increase in the bicarbonate due to, we'll now say hypokalemia, is a transcellular effect. It's not an effect on the kidney or any other organ. It's not a vein per se, unlike most of the causes of metabolic, or most of the factors. This is generating bicarbonate, but it's not increasing the total body bicarbonate. It's just shifting bicarbonate when you look at the stoichiometry, and I'll show that in more detail here, from the cell to the outside. It's actually not bicarbonate that's moving, it's a proton. So we have a transport processes in the cells that can either exchange potassium, so the potassium comes out in exchange for a proton, which is what we're showing here. The potassium can also exchange for sodium. But remember, the potassium in a cell is about 100 millimolar. It's way greater than your blood. Your blood is around 4 millimolar. The potassium is 100, 110 inside a cell. Sodium in a cell is only 20, 140 outside. So the concentrations are very different. But if you have potassium coming out of the cell, to maintain electroneutrality, you have to have another cation coming in, or the potassium, I guess, could leave with chloride or bicarbonate or some other organic anion. But this process is what we're about today. So the potassium comes out, and if it exchanges for a proton, you actually generate one bicarbonate ion in your extracellular fluid. So you alkalinize the extracellular fluid, you acidify the intracellular fluid. pH in a cell is normally 7.2. It's not 7.4. So the pH in the cell gets lower, the pH outside gets higher, bicarbonate outside gets higher. And in effect, you've transferred one bicarbonate ion from the cell to the outside. So you haven't done anything with total body bicarbonate, like you do with vomiting, where you have a proton coming into the lumen of the stomach and you have a bicarbonate ion leaving the parietal cells into your gastric vein. There, your total body bicarbonate content, in quotation marks, goes up. Here, total body bicarbonate's the same. You're just shifting it from one compartment to the other. But this is now felt to be the mechanism by which hypokalemia in someone with metabolic alkalosis, or extra bicarbonate, whole body input-output kidney mediated mechanism. So this is very interesting and important to memorize. Now, if you just have dietary potassium deprivation alone, very simple approach, you put an animal or a human on a low potassium diet. Again, it's not so simple when you think about it, because you can't just lower potassium in the diet. It either has to be potassium chloride, or you have to do a potassium sodium swap, or you have to lower potassium phosphate. You can't just remove one ion. But the way they did it was a few ways. But at any rate, potassium deprivation on a normal sodium chloride diet, you hardly see any metabolic alkalosis. And the thought is you don't get much HK exchange. You don't get much transcellular shifting. However, if you put the patient on a low sodium chloride diet and a low potassium diet, you get a much higher blood bicarbonate level. And probably that's occurring because the sodium concentration drops, or there's less sodium, and you don't get as much potassium-sodium exchange. So you, on a low sodium intake, you actually enhance potassium proton exchange. Remember, the potassium can exchange for proton or sodium, or come out with chloride. But on a low sodium chloride diet, what you're doing is you're enhancing the HK exchange mechanism. So remember that. If you have a patient who's sodium depleted and K depleted, those factors alone are going to significantly raise the blood bicarbonate concentration and be an important factor for the generation of a metabolic alkalosis, but it's not a vein. It's most cells in the body can do this. So we'll check off here hypokalemia and K depletion as, and again, I don't want to make a major distinction between these. It's just hard to invoke mechanisms as to how input-output problems or total body K content could lead to anything. I mean, it's just, what are these sensors, or how would that signal even be known by the cells? Cells measure concentrations, so respond to concentrations. And so this is most likely what's going on. Although, as I say, someone needs to do an experiment where they make hypokalemia without K depletion and vice versa and see what happens. It's just never been done. So we'll check those off. They're hypothesized, but there's enough data to show that this is correct. And again, we're checking off sodium depletion, not because sodium depletion per se leads to an elevated bicarbonate. If you just put someone on a low salt diet, nothing's happening. Their blood bicarbonate's not going up, but it's connected to the hypokalemic effect, because sodium depletion in the presence of hypokalemia diminishes the sodium potassium exchange mechanisms in the cell and enhances the proton K exchange mechanisms. Mineralocorticoids, you'll see this everywhere. Patients with primary aldo, patients with other mechanisms for aldo going up. But if you do the experiment rigorously, giving mineralocorticoids hardly changes the bicarbonate at all. However, if the patient's also K depleted such that they become hypokalemic, and you give them mineralocorticoid, you're going to get a much higher blood bicarbonate concentration. So the K is not only coupled to the sodium depletion, the K is also coupled to the mineralocorticoid effect. And obviously, we're separating these things artificially. They could all be occurring simultaneously. You can see how the matrix just gets more and more and more complicated as far as the cause and effect phenomenology. So again, if you give mineralocorticoids to a human or an animal and keep the K constant or have very little change in potassium, nothing happens. However, if you lower the K, you get a much greater effect. It also was found years ago in the 1950s, actually, at Yale, that if you have decreased sodium chloride intake, then the mineralocorticoid effect decreases. So these things are hard to predict. You'd have to memorize them, unfortunately. So a lower sodium chloride intake ameliorates the mineralocorticoid effect, whereas potassium depletion enhances the mineralocorticoid effect. Mineralocorticoids alone do nothing. So we'll check off aldosterone. We've already checked these off. Remember, these are now coupled with each other. And the aldo and the sodium depletion are coupled to each other. If you're sodium depleted, you get much less of an aldo effect. Now, what about chloride, which we're going to talk about for the rest of the talk? In the previous weeks, we talked about how in the studies that were done, chloride delivery to the distal nephron was felt to be an important mechanism as to how chloride is affecting the blood bicarbonate level. But remember, the word distal nephron, which, by the way, you should not be using anymore as a renal fellow, because there's 9 different tubules in the distal nephron. And I think I'm going to give a talk to you on the anatomy of the nephron. I mean, it's funny. I mean, residents and medical students know the coronary arteries, but I think it's very important as a renal fellow not to keep talking the same way as you did before, because when you're saying distal nephron, it will be analogous to saying you're living in West Los Angeles. You're not really saying anything. So there's 9 different tubules, and they all have different cell types that all have different transport functions. And to really understand the clinical diseases and particularly acid-base disorders also that you're looking after. You really need to know the anatomy and the different cell types and what they do. And it's hard at first, but you get it eventually. It becomes repetitive. So at any rate, at this time, no one knew the different segments. And this was done in the 80s. The knowledge of the nephron really wasn't what it is today. And so, but they made the distinction between the earlier parts of the nephron and the distal parts of the nephron. This was a hypothesis. No one had ever, this was a hypothesis based on the experimental data where they just did whole kidney or whole animal balance studies. And from clues that they saw, which I won't get into today, they hypothesized it was the distal. There weren't experiments showing it. And it didn't appear to be the blood chloride concentration. They varied the chloride. It really didn't affect the bicarbonate. It didn't look like sodium balance was involved. And volume expansion per se, or in other studies, depletion, independent with a constant GFR, with a constant GFR is the key word here, did not have an effect. So as long as you kept the GFR constant but changed the extracellular fluid volume, they did this. I don't know if you remember, I talked about those experiments with aortic clamps where they could clamp the aorta to keep the GFR what they wanted it to be and change the extracellular fluid volume independently. As long as the GFR was kept normal, the volume expansion had no effect at all in ameliorating the blood bicarbonate level. However, GFR is important. And as I talked about before, Marty Kogan at UCSF years ago showed that it is important in the maintenance phase. And if you want to take this to the clinical level, if you give someone with CKD5 a bicarbonate load, and you give the same exact load to someone with CKD2, the difference being their GFR, you're going to have a much more severe metabolic alkalosis with the CKD5 patient. They just don't have as much bicarbonate coming through all their glomeruli per unit time. And so it is important as far as bicarbonate loads in patients with impaired renal function as a sort of a simple model. You could think of it like that. So GFR is an important factor. And obviously, if you have someone with ESRD with no GFR, the entire bicarbonate load is going to stay in their body, and they're going to have even a greater elevation of their plasma bicarbonate level. So for a given input, GFR is important, independent of potassium, sodium, chloride, everything else we just talked about. Now, what about the role of chloride? So people, hypothesize that it's somewhere in the distal nephron, but still no one really understood exactly what was going on and whether it was chloride delivery or chloride concentration. Exactly, they knew chlorides, they knew the words distal nephron and chloride, and that was about it. So Gala and Luke really did a lot of studies in the late 80s and mid 90s looking at this in more detail. And in this paper, they looked at the importance of chloride for correcting the metabolic alkalosis in the rat. In their model, they had the rats on a low sodium chloride diet and actually peritoneally dialyzed the rats, exchanging chloride for bicarbonate in the dialysate to create the metabolic alkalosis. So they raised the bicarbonate in the dialysate, lowered the chloride, and they created this transient metabolic alkalosis this way. And then therapeutically, they gave the animals glucose to drink, choline bicarbonate. Choline is a monovalent cation. It can replace sodium. So no sodium, but bicarbonate, or choline with chloride mixed with choline bicarbonate. And it turned out that only the group that got chloride in the drinking water corrected. Getting glucose didn't do anything. Giving choline bicarbonate didn't do anything. And also notice here, there's no sodium. They gave non-sodium salts, and the metabolic alkalosis corrected. And in their studies, it corrected without an increase in blood pressure, no change in GFR, plasma volume, or sodium balance. And also aldo didn't seem to be required either in the maintenance phase, and the decrease of aldo was not required in the correction phase. And for the first time in the literature, they came up with this term called chloride depletion alkalosis. The term is not great because it has nothing to do with the delivery of chloride to the we'll still use the word distal nephron, even though I hope at the end of the talk you won't anymore. But it has nothing to do with chloride depletion per se. So why they use that term is not clear. And then they did another paper where they looked at the effect of chloride and extracellular fluid volume on bicarbonate reabsorption in the nephron. And for this, They did the same protocol, the peritoneal dialysis exchange of chloride for bicarbonate to generate the metabolic alkalosis, and they were looking at the effect of chloride and volume. And volume, they were looking at by giving 6% albumin expansion to the animals. So the animals either got that or a 0 sodium chloride containing solution. So there's a lot of numbers here, but we'll just focus on the green and blue initially. This VE group is the albumin group, and the bicarbonate was 40 after getting the albumin. It didn't do a thing. In the chloride-containing group, it dropped, from its initial level, which was higher, to 34. So the chlorides seem to make the difference. And I won't get into the other numbers, but they showed that the metabolic alkalosis persisted in the rats getting albumin despite significant ECF fluid volume expansion. So this again just drove home the point that volume expansion has nothing to do with ameliorating the metabolic alkalosis, with the proviso that the GFR is not changing, because the GFR alone can, as I talked about with the CKD patients and Marty Kogan's animal and human studies, that GFR increasing can correct metabolic alkalosis, and GFR decreasing contributes to the maintenance. But with the chloride-containing solutions, despite the fact that they were still volume depleted, actually, the animals were still volume depleted, but just giving chloride corrected or helped correct the metabolic alkalosis. So again, there's something unique about chloride. And then Rosen, a year later, continued to look at the effect of chloride and volume, and they had a different protocol. And this is a human study as opposed to these other animal studies. They gave Lasix, they gave sodium K citrate. Remember, citrate's converted into bicarbonate. When the carboxyl groups are neutralized, just like ketone bodies or lactate or acetate in TPN, all these organic anions are converted into bicarbonate with a lower sodium diet. And they found that, and they did two sort of corrections. They gave either potassium chloride or they gave sodium phosphate. It's a bit of a messy study because there's all these different ions, but they found, you can see here that they generated the metabolic alkalosis. This is the total CO2 concentration from the metabolic panel from 27 up to 33. They got a nice maintenance phase for a few days. And then they gave the KCl, and you can see the bicarbonate coming down. Again, no sodium. But when they gave the sodium with phosphate, nothing happened. So chloride, in this study too, you can't rule out potassium, but seemed to be what was going on here. But they hypothesized for the first time something different. And I'm not showing that data that had never been hypothesized before. They hypothesized that the chloride effect was mediated by the distal nephron. But for the first time in the literature, they said that there's some chloride bicarbonate exchange process. They also said it could be a chloride proton symport process. So you can't really distinguish these, at least in these kind of studies. But the word chloride bicarbonate exchange as the mechanism in the distal nephron appeared for the literature in the first time. But the question again is where? The distal nephron, as I said, has 9 segments, again, not known at that time, but which of these segments is causing the effect? Given that there are these segments with totally different cell types. But just showing pictorially here what they hypothesized in this very prescient paper, that somewhere in the distal nephron shown here with this cylinder, there was a cell or a process secreting bicarbonate into the lumen and absorbing chloride. The secretion of bicarbonate would ameliorate the metabolic alkalosis, and the absence of chloride in the lumen would stop this from occurring and contribute to the generation and maintenance. So the thought is that bicarbonate is secreted in some cell somewhere in the distal nephron, but it's a chloride-dependent process. And when you're chloride depleted or you decrease the chloride delivery, the luminal chloride concentration drops and shuts this thing off. Now you can't secrete bicarbonate anymore. So it would build up in the blood. That was sort of the hypothesis here that appeared really for the first time in the literature. So Luke and Gallo went on to try to figure out where in the nephron this is occurring. And what they did is a procedure called micropuncture, where you stick pipettes into the nephron. You can only do that in the superficial nephrons. in certain types of strains of mice. So they stuck a pipette in different portions at the tip of the thin descending loop of Henle here, which would be deep in the inner medulla, the papilla, and then way up here in the cortex near the distal convoluted tubule. But the point is that they could, and then they looked at the final urine here. So what they wanted to know is, what is the bicarbonate flow in the nephron? If they could find out a difference between these pipettes where bicarbonate was increasing significantly, that must be due to bicarbonate secretion. And they did their peritoneal dialysis model again. They created a metabolic alkalosis, and then they corrected the animals with 0 sodium potassium chloride solution or their dextrose solution. But what you see here is something interesting. When you look at so the flow is going this way. This is the glomerulus. It's coming down here. First hits the thin descending limb, and then their pipette can measure up here in the cortex. So they wanted to see, is there any difference in bicarbonate delivery? So this is amount per unit time between the tip of the thin descending limb here and here. If there was, that means bicarbonate must have been added between the two. And they found it was the same. They really didn't find any difference between these two sites. But when they compared this site to the final urine, remember, the flow is going this way. So when they compared the distal convoluted tubule to the end urine, there was a big difference. Big difference. So what they hypothesized was that it's occurring, yes, indeed, in the distal nephron, but where? Totally unknown. Well, other studies were being done before that led some clues as to what might be going on. This study looked at bicarbonate transport in a part of the nephron called the cortical collecting duct. This shows a little blow up of the nephron, and some of the segments in the distal nephron are shown here, the connecting tubule, the cortical collecting duct. The rest are really not shown here, but there's 9 total going along the collecting duct here. And they studied the cortical collecting duct. The procedure they used was called in vitro microperfusion. It's something I did at the NIH, where you take a tubule, you dissect a tubule, which takes you about a year to learn how to do, and then you couple it to 2 micropipettes, and you can actually put fluid down it and study transport across the epithelium. Mo Berg created this procedure in 1963, actually. People had done this with neurons. Everybody told me, you're crazy. He was just a postdoc. That the tubules are going to die. You'll never be able to study them. Anyway, he really changed nephrology, and we know basically everything we know about whole tubule transport from this technique, not from micropuncture, because micropuncture, you can't get all the nephron segments. With this technique, you can get any segment. And this just shows a blow up of the cortical collecting duct, and you're actually looking in the lumen. So imagine a cylinder that you've cut, and you're looking at cells on the bottom. And it's a very interesting segment because it has at least 3 different cell types. There's this kind of cell here. This is called the principal cell. Each one of these cells has 1 cilium. I don't know if you can see it, and it's in the lumen of the tubule. People think this is a flow transducer. It actually measures the luminal flow rate, which is, it's very small. It's in nanoliters per minute. So, 10 to the minus 9 liters a minute are the units we're talking about. But the cell is able to measure the flow and change its transport properties. This is the cell that has ENaC in its apical membrane. So the ENaC channels are sitting here. Amiloride binds here. That's how amiloride works clinically. It binds to the principal cell. And aldosterone binds on the opposite side, on the blood side, on the mineralocorticoid receptors. The acid-base part is taken care of by what are called intercalated cells. They look like they have these worm-like, they're called microplici, and they're on the surface, and the intercalated cell has 2 types. One cell is absorbing bicarbonate because of proton secretion. This is the cell that has vacuolar proton pumps that are mutated in patients with distal RTA. And then the other cell is now felt to be the bicarbonate secreting cell, but none of that was known at this time. What he found was when he gave the animals sodium bicarbonate, these tubules secreted bicarbonate. So everything below 0 is secretion, bicarbonate coming into the lumen from the outside solution. Whereas when he gave the animals ammonium chloride and created a metabolic acidosis, the tubules absorbed bicarbonate. This just blew the country apart. People who were interested in this, no one had ever seen anything like this before, where one tubule could change totally its direction of transport based on the systemic acid-base balance of the animals. And of course, no one knew the cell types or what was going on. All they were measuring was direction of transport. With sodium bicarbonate load, they were secreting bicarbonate into the center here from the outside, and the animals that had a metabolic acidosis, they were absorbing bicarbonate, and the animals that had normal acid-base balance, they were close to doing nothing. Now, that could be that they weren't doing anything, or there was secretion and absorption that were going on at the same time that were opposite in direction, giving a result that's about 0. But again, these are just thoughts afterwards. No one knew what was going on then. And then further studies were done. Now that they knew that they had bicarbonate secretion, they can now look at the effect of luminal chloride in the isolated perfused tubule, where you have easy access to changing the chloride concentration in the lumen of the tubule to whatever you want it to be. And you could look at, in those experiments, bicarbonate secretion. So this was done in the 90s, 1990, a little later. And so first, the model of metabolic alkalosis was created. And you could see here that in the controls, these animals were absorbing bicarbonate. It wasn't close to 0. It depends on the animal. Rabbits tend to be more close to neutral, normal rabbits. Rats tend to have a bicarbonate absorptive net process, but they could also be flipped to secretion with metabolic alkalosis. So what they did was they took these tubules that were secreting bicarbonate. Remember, the negative direction is secretion here. Okay, it's just arbitrary. It doesn't have to be, but just secretion is here, and increasing secretion is downward, sort of opposite of what you would intuitively expect. And then they remove chloride from the lumen, and look at that, the secretion stopped. And then when they put chloride back, the secretion came back. So they showed for the first time, they confirmed there's a bicarbonate secretory process, but they showed for the first time it's dependent on the luminal chloride concentration. And then things started making more sense with what I showed you before, this chloride delivery and the reliance of the maintenance and the correction of the metabolic alkalosis on chloride started making more sense with the thought that people started now zeroing in on the cortical collecting duct and its bicarbonate secretory capacity, and the fact that it seems to be entirely chloride dependent. They also showed, and this isn't so important for the talk, but they also showed that the animals that had a higher serum bicarbonate had a higher secretion, suggesting that either the bicarbonate or the metabolic alkalosis was driving this process more and more in a dose-dependent fashion. They also looked at chloride balance and found the same sort of thing. But again, chloride balance might just be a reflection of the luminal chloride concentration. So for the first time, people could now say, yes, not only does the cortical collecting duct secrete bicarbonate, but it appears to do so via chloride bicarbonate exchange process, shown here, where you have some sort of cell. So we'll take it from the word process to a single cell, simplistically, just for argument's sake. And this cell secretes bicarbonate from the blood into the lumen of the cortical collecting duct, but it does so in exchange for chloride. So that has a number of implications. The main one is that if you remove chloride from the lumen, the thing stops. If you enhance the chloride concentration in the lumen, you stimulate it. And same thing here. If you enhance the bicarbonate concentration in the blood, like was shown, the worse the metabolic alkalosis, this thing will go faster. So for the first time, people then started putting things together that it's the cortical collecting duct and there's some chloride bicarbonate exchange process. Now, other things were occurring simultaneously in the literature that then got coupled to this. And the gene encoding the protein that is doing what we just showed was found serendipitously. In Vaughan reported 2 children with congenital deafness and goiter that is now called Pendred syndrome. And Everett in 1987 identified the gene and mutations in it. And the gene is called pendrin. It's SLC26A4, the gene name. Scott in 1999 showed that pendrin is a chloride iodide exchanger, which is now thought to mediate iodide exit from the thyroid. And somehow, when this transporter is mutated, you get what's called Pendred syndrome. And it also looks like it's in the ear. That's why you need it to hear. And that's why you get in the cochlea, that's why they're congenitally deaf and they have a goiter. In 2001, people started looking to see where else this thing might be. And in addition to the thyroid and the inner ear, pendrin was found to be expressed on the apical membrane in the type B intercalated cell in the cortical collecting duct. CCD is just same thing as cortical collecting duct. And here's immunocytochemistry showing pendrin staining of the, so this is the urine side, the urine side here. This is the cell. This is the blood side. And on the urine or luminal side, pendrin is staining the type B intercalated cell. Aquaporin 2 is just a marker for the principal cell that I showed you before that has a cilia. And this is what amiloride is binding to this cell. So pendrin seemed to be on these cells. Now, to prove that this cell was the cell secreting bicarbonate, they looked at pendrin knockout mice, where you delete the pendrin gene, and compared it to the wild type mice and looked at JTCO2 means bicarbonate secretion in picomoles per minute per millimeter. It's corrected for length, but it doesn't matter. This is the amount of bicarbonate being secreted. It's in the negative direction because it's secretion, not absorption. And they just did the simple experiment. These are cortical collecting ducts. What will happen in the knockout mouse? So in the control animal, they were secreting bicarbonate, but in the knockout mouse, it stopped and they were left with bicarbonate absorption. So this proved that pendrin was the protein that was causing the bicarbonate secretion and then the chloride bicarbonate exchange. Other studies went on later to show that pendrin isn't just a chloride iodide exchanger, it also can exchange chloride for bicarbonate. So from these studies, we could see now that CCD, the cortical collecting duct, secretes bicarbonate via pendrin. So this protein here, or cell, you can think of it either way, is pendrin, and it secretes one bicarbonate ion into the lumen of the CCD in exchange for chloride. And this is the part of the so-called distal nephron we're talking about. But as I said again, there's 9 segments here. The cortical collecting duct just comes after the connecting tubule and is sitting way up here in the cortex. So this is cortex, outer medulla, outer stripe, inner stripe, and the inner medulla that's divided up into 3 different sections, not shown here. So we're dealing with something which is high up in the cortex. That's where humans and all animals secrete bicarbonate. Nowhere else in the nephron does this occur. Now, other studies were done trying to look at the importance of pendrin in normal animals versus perhaps stimulated with mineralocorticoids, or et cetera. So the idea was here, let's look at the systemic acid-base status, not fancy studies like in vitro perfused tubules or measuring ion delivery within the lumen of the tubule, but let's just look at systemic acid-base balance. And what they found was that if you do look at the knockout animal, the knockout mouse, the acid-base balance is no different. The black is wild type, the hatched is knockout. You can see that the pH, PCO2, and the calculated bicarbonate are no different, whether you have pendrin or not. So in the knockout mouse, it doesn't affect your systemic acid-base balance. So the thought was, under normal conditions, it's probably not that important, or the animal can compensate. You know, that's always unknown. And that's why when you do a knockout, there are knockouts that are called conditional knockouts. If you do the knockout from birth, you're likely going to get compensatory mechanisms that will ameliorate the effect you're trying to see. Whereas if you do a conditional knockout, you can do that in the adult and you can study before, during, and after. It's like giving a Lasix or something. You study before, during, or after. You might see something. So it's unknown whether, in an absolute sense, the effect wasn't that important. or the animals had compensated. As far as I know, no one's done a conditional knockout of pendrin. But the animals that had gotten mineralocorticoids, you can see that in the absence of pendrin, the metabolic alkalosis was much worse. So presumably under certain stimulatory conditions, maybe hypokalemia, maybe from this study, mineralocorticoids, or other factors that can enhance the chance of getting a metabolic alkalosis, maybe then pendrin turns on, and without it, you can see the effect of the metabolic alkalosis much greater. So the question then, and it's still unclear, is does pendrin regulate acid-base balance in humans? In 2008, there was a case report of a patient with pendrin syndrome who developed a very severe metabolic alkalosis. You can read that article. It's an interesting one. After just getting thiazides. And, you know, thiazides can cause a mild metabolic alkalosis, but not a very, very severe one. So the thinking from this case was that pendrin was turned on and was dumping bicarbonate in the urine as much as it could to keep the blood bicarbonate from going up. And remember, with thiazides, you've got chloride in the urine, so the pendrin can work. It's not like someone vomiting or someone who's volume depleted, where the chloride concentration in the urine is low and shuts pendrin off. In this type of model of metabolic alkalosis, there's chloride in the urine. So presumably, so the thinking was that pendrin was ameliorating normally this, but when the thiazides were on board, it was much worse, and it was corrected by giving spironolactone and potassium chloride. Again, not very definitive, a lot of hypotheses involved. You can't stake any definitive interpretation on this, but it had some interesting implications. And then in 2011, there was another case of pendrin syndrome with a very severe metabolic alkalosis just after vomiting. And again, the vomiting didn't seem sufficient to cause that severe elevation of bicarbonate. So again, the question is, when you have pendrin syndrome and you lose this transporter, perhaps all things that cause metabolic alkalosis are going to be exacerbated. And then in 2015, pendrin expression was studied in patients with distal RTA and was found to be decreased. So it was a cute study, not hard to come up with other hypotheses, but one thought is, well, maybe pendrin, decreased pendrin expression was a result of the distal RTA, and that's what you would want. You'd want to decrease bicarbonate secretion. If you already have RTA or a metabolic acidosis, you would want, when you have a metabolic acidosis, you don't want to have pendrin working, because otherwise it's just going to get worse. You're just dumping more bicarbonate in the urine. So this seemed to be an adaptive response of the kidney, maybe, to help ameliorate the drop in the bicarbonate. And then in 2016, exosomes containing pendrin. I don't know if you've heard of exosomes, but if you look at any urine, there's little parts of the membrane that come from different nephron segments that you can actually look at and study in the urine. And these membranes, they're sort of like, they're circles, they look like vesicles. They contain transporters from the nephron. They're parts of the cells that break off all the time. So the urine has this big cellular component that we don't think of clinically, but when you're doing studies, you can isolate these exosomes. You can study the proteins that are in them, either with mass spec or with immunocytochemistry, different ways to do it. And what they found is that there's pendrin in these exosomes, and that after ammonium chloride loading, which again causes a metabolic acidosis, the exosomes containing pendrin decreased, as you might expect, if there's less in the membrane, as was shown in the distal RTA patients. And the exosomes containing pendrin increased after sodium bicarbonate administration, which you would think teleologically would occur because the nephron is trying to dump bicarbonate, and it does so by increasing secretion with pendrin. So we're now going to have to check off the luminal chloride. And in fact, clinically, this is probably the most important factor that is related to the maintenance and correction of metabolic alkalosis. So when your attendings start talking to you about metabolic alkalosis, or when you're studying it for your exams or thinking about it, focus on what the luminal chloride is in the cortical collecting duct. because if it's normal, you've got pendrin working normally, most likely. If it's low, you're not going to be able to correct the metabolic alkalosis unless you give chloride. That's what giving chloride does. Giving sodium chloride or choline chloride or K chloride, whatever chloride salt you want to give, is allowing the cortical collecting duct to have chloride in the lumen. And that will allow pendrin to secrete bicarbonate in exchange for chloride. But if the chloride in the lumen gets too low, like a vomiter or other patients who are volume depleted, who have avid chloride, remember, there's avid chloride reabsorption in the proximal tubule and in the thick ascending limb in those patients. By the time the urine gets to the collecting duct, a lot of the chloride's been absorbed. Unless you give IV chloride or oral chloride, you're not going to be able to get pendrin turned on. So the take-home message is when you're giving chloride, you're not volume expanding the patient. You're not doing anything importantly physiologically other than raising the luminal chloride in the cortical collecting duct, allowing pendrin to work and dump the bicarbonate. Chloride depletion, as I say, I'm not checking off because there really is no way of interpreting how whole body chloride loss could send a signal to some sensor in the kidney or something and cause it to change bicarbonate transport. So I'm leaving that off. And I think I'll stop there today. Next week, we'll talk about the tubule transport machinery. We've talked about one transporter, pendrin. There's a lot more involved in what's going on, but today I wanted to take it from sort of the growth, the larger level of looking at the earth down to at least the house that we're talking about. So remember pendrin and remember its important transport of chloride and bicarbonate in your patients. And I'll stop there for today. So thanks very much for listening. Does anyone have any questions or anything that wasn't clear? Okay, everybody, have a great day. I'll stop there for today and we'll see you next week.